So I decided for the title Microbial Networks. I was not aware that Vittorio is so interested in networks, but I hope that I can explain a lot of these networks here during my talk. And I have two affiliations. So one is our university. This is Graz University of Technology, and the other one is the ASIP. So this is an Austrian Center of Industrial Biotechnology, because I'm in the Faculty of Chemistry, Engineering and Biotechnology, and we have this center, and this is open for cooperation between industry and university, and for us it's very good to have this second possibility for cooperation. And so I will start. Yeah, this is my group. So I have 30 people, but a lot of students for sure. And not so many people are paid by the university directly, so most of them have an external funding. So here is Graz, so it's not so far away from the border and not so far from Trieste. And the institute is Environmental Biotechnology, and we try to combine basic research on microbial ecology. So I'm an ecologist and microbiologist with biotechnology, and with a specific focus on the plant microbiome and on biocontrol. But today we also moved a little bit to the human microbiome, because it's so interesting also to see the parallels, and I will show you also a little bit of these research, because normally it's much more interesting to learn something about ourselves than from plants, but I will do both. So yeah, here this is the institute in the back, and this is the group. It was from last year, the picture. And we have also two startups at our institute, which is really nice to have them. One is Biotense, and this is more like a think tank, so they have knowledge in formulation of bacteria, for example, for seed application, but now we're also working on fecal transplant to develop a formulation for specific donors for fecal transplant for risk patients. And we have another one, this is Room Biotic, and they work on the indoor microbiome to find, on one hand, new ways for sterilization, which is important for several things like intensive care units and also for the production for pharmaceutical approaches. And on the other side, we are also working on indoor microbiomes, because they are very important for our health, and a lot of diseases come from these problems with the indoor microbiome. Yeah, this is the background, and now I will move to research. Yeah, due to all these new techniques which we have, next generation techniques, all these omic techniques, but also novel metabolomics approaches, and microscopy is also very important now. We see that all the eukaryotes are more or less holobionts, or meta-organisms as we call them, and they act in many things together. And for the plant microbiome, for example, a lot of for plants, a lot of bacteria allow germination. So there are several branches of plants, for example, orchids or bryophytes. They cannot germinate alone, so they need really specific microbes for germination. Also, plant growth, health and performance is strongly correlated with the plant microbiome. So, and you know, there are much more genes and much more cells available from the microbiome than from the plants themselves. It is similar to us. So our microorganisms have a weight of 2 kilograms. So, and this is like the brain, and it's very important for us, and it is very similar to the plants. So, and we realized that this networking between the plant and the microbiome is very important, and at least all diseases are associated with imbalances, or as this comes from medicine, they called it dysbiosis. And this is my introduction. So, and today I would like to give you some examples for these networks, how it works, how specific they are, and then I would also like to show you a little bit about the connection to other microbiomes, and in the third point I would like to show you some solutions how we can avoid these dysbiosis effects by biocontrol. So these are my 3 points. So first I will give you several examples. I selected 3 examples for the network analysis, and then I will give you also an overview about our research on this. And then here, this is you can see it as an excursion to other environments. This is only a short part, and then the last part is biocontrol, focus on biocontrol stages. So I would like to start with mosses. So 10 years before, we started with the moss analysis because these are the oldest land plants which exist on Earth, and we thought they should have a very good relationship to their microbiome, and therefore we selected them as model organisms to understand plant-microbe interaction. And this is really a basic project. We have here the Austrian Basic Science Foundation, FWF, who supported this project. So I started it here in Rostock at the Baltic Sea. So for many years I lived there before I moved to Graz, so 10 years before. And the northern hemisphere is covered by these bog ecosystems, and also Russia. And here, these are my Russian colleagues, and this is Anastasia, and she was a project leader of this one. Yeah, and as I told you, these are living fossils, and when we started, there was more or less no knowledge about the microorganisms, but we thought that especially in these ecosystems, so these bog ecosystems, they are ombotrophic living rooms, so that means that they live only from the rain, so they need a lot of support from their microbiome to exist and to grow. And as I told you, since 10 years now we study these ecosystems, and by the way, they are also very important for our climate because they are the most important carbon sink sources. So also the alpine bogs, so they are very, very important, and these are the first ones which are affected by climate change. So here you can see a leaf of a sphagnum, and you can see that there are a lot of bacteria inside. So this is a specific probe for archaea, therefore there are not so many. And this was also interesting because, as I told you, we had this project together with Russians, and we found a very, very high similarity between the microbiome of the Austrian box and the Russian box, although the plant specificity was very high. So these sphagnum plants, there exist 300 different species, although for us they look all of them looks like the same, but the biologists, they can differentiate them, and we found a highly similar microbiome so far away, and therefore we thought there should be a mechanism behind why we have this similarity, because they are very old, and how can it be that it is so similar? Partly we found the same clones, and then we developed the hypothesis that maybe from one generation to the other they will give a core microbiome to the new moss generation. And here we have a haploid generation of the gametophyte, and then we have the sporophyte. This is a diploid generation of the plant, and indeed we really found that associated with the spores inside of the sporophyte, here is the moss and here's the sporophyte. You can see here a high diversity. I will not go into detail into that, but they really give their new plants an inoculum, and surprisingly, all these which we found here in the core microbiome, they are well known for nutrient supply, so for nitrogen fixation, phosphorus solubilization, and so on. So at least it's like humans, it's a parallel, because here you know that during birth, so we also, the mother gives the newborn an inoculum, which was for a long time, it was ignored. And by the way, so I think Italy is has one of the highest proportions of Caesar section, which is really not good for the babies, because then they get the microbiome from the hospital indoor microbiome, which is normally characterized by a lot of multi-resistant pathogens. So going back to the mosses, learning from the mosses and give natural birth here to the younger people. Yeah, this is an insight into the capsule of the mosses. Here are a lot of bacteria in red, and this was the first evidence that also the plants can give these core microbiome to the new generation. Now it is also known for seeds and for other plants, but due to the huge diversity of plant families, it is specific for each plant family. So the plants develop specific strategies to handle their microbiome, but the mosses, so they are so intelligent to do this. Yeah, and here again a picture from a moss leaf. You can see here very nice colonies inside of these cells, and here you can also see that they interact with each other, so it would be nice to have here some complementary genomics. And what we have done here also for our sphagnum microbiome, we sequenced the whole metagenome, so including all functions. So we isolated it, and then we sequenced all the genes which are associated here. We found 1,000 different species, so it's similar to the gut, so it's high diversity. And so the first point was that we got such an amount of data that we destroyed one of our TU server, so which was not expected that it comes from us, because we have a huge faculty of computer science and normally they handle a lot of big data, but we had more data for one sphagnum microbiome than all our computer specialists. But at the end we were able to handle it also with help from outside, but you really need a lot of capacity to assess those things. So, and then there's a MGRust server, which is very nice. It's provided from the US, and then you can compare your data set with all the other metagenomes. So if you publish anything about a metagenome, you also have to put it into such a public server as MGRust, but it's at the moment, so they have also problems because to pay for the storage is very difficult. So, but you can give it to China or to the US. And so you can see here our microbiome is in between the plants and the fresh water, which makes sense because these boxes are also covered by water. Here are the human microbiomes, which are in MGRust. So you get an unbelievable amount of data, and you can assemble all of them and make some pathways for nitrogen fixation and for everything. So, and before we started this analysis, so this was Anastasia, a postdoc for 3 months. She only was inside of this data set. So we developed some hypotheses about the function, about unique functions of our microbiome. And we thought they should supply a lot of nutrients, and they should be also responsible for pathogen defense, because these bog ecosystems, they are monocultures at low pH, and normally there should be a lot of fungal pathogens inside, as in our agricultural fields, but they are not known. In contrast, So these mosses, especially these sphagnum mosses, are well known for traditional use in antifungal activity. And here also we used it, or our ancestors used this as natural nappies. So in Europe it was widely used, and the last use was during World War II. So the soldiers used it for wound covering, because it has this water holding capacity on one hand, and on the other hand this antifungal capacity, which was really nice against fungal infections, for example for babies, for candida, against candida infection. So, and we found some here, this is a comparison of our metagenome with all functions in comparison to all the other metagenomes, and we found some of our hypothesis could be confirmed. So for example, they provide a lot of nutrient, and they have unique features to avoid stress tolerance against abiotic and biotic stress. This was nice, but we found also several surprising things, because I thought this is a very old ecosystem and it should be well developed, but we had a higher interaction via quorum sensing than all the other metagenomes. So it seems that they exchange a lot and talk a lot. And in parallel, a lot of horizontal gene transfer and a high number of mobile elements, which I had not expected before. Yeah, this was a metagenome. And now we performed a metagenome on the whole bog ecosystem, so including also all vascular plants. And so then here the grasses, mosses, peat mosses, and also shrubs, Pinus mugo, and all these vaccinium species are inside. And then we calculated a core microbiome. And at the end we found out that the whole bog is connected by these sphagnum mosses, which is clear because whenever you have seen any bog ecosystem, so it's dominated by them. And the bacterial keystone species is Burkholderia. So Burkholderia is here completely in the core. So a lot of these, from the plant-associated group of Burkholderia, we found a lot. This is the most dominant, and it occurs on all bog plants. So on the first level of the plants, sphagnum is a keystone species, and on the bacterial level, Burkholderia is the keystone species. So coming from mosses to sugar beet, and now we are, by the way, here are a lot of wild sugar beets, so we also have investigated those, and it was also interesting to see that they lost a lot of diversity from this natural here place on the Mediterranean Sea, so that the domestication really, it's also a breeding of the microbiome, but it is indirectly, so they do not know that. So, and this is the last example. This is a lettuce, and we had a project that was funded by the EU, and is together with our other university in Graz, and also with Rita Grosch in Großbeeren. And yeah, here is a rhizosphere colonization, and you really can see that there are a lot of bacteria. And this is work done by Massimiliano, so we have always some Italians in our group. And what I don't know is, before we started this project, is that lettuce is a very, very old plant of mankind. So also in old China, so they breeded a lot of lettuce, and in Egypt, and it is very difficult to follow the breeding lines of lettuce. So I thought it's more modern to eat lettuce, but it's not true. And there exist so many cultivars worldwide, and it's very difficult to get an overview. But fortunately, we had this cooperation with Arche Noah. This is in Austria. It's not a company. It's a group of people associated with Boku, and they try to maintain all the cultivars, and they also cultivate them into the field. And here we selected 8 different cultivars, and the wild type, this Lactuga cereola, you can find it as a weed outside. And we compared these 9 plants, species and cultivars. And here we analyzed, Massimiliano analyzed the rhizosphere, and surprisingly we found 50% is a core, and 12% are really cultivar-specific bacteria. So, and this shows that breeding really influenced the microbiome of us. Not of us, first of the lettuce plants, but when we eat, then also our microbiome. And yeah, and using here all these weeds which we had, we performed a network analysis. It was published by the Knight Group 2011. So you need a lot of replicates, and then you can perform this network analysis. And we compared it with all the other networks which were published for microbiomes. And we found that especially lettuce has a very, very loose structure. And our conclusion was that maybe this explains why lettuce has very often pathogens, but it is also very good for biocontrol. So if you look into the literature, and also our own experience is that biocontrol in lettuce works very well. And maybe this could be explained due to this loose network structure, which allowed other species to invade into this network. So, but it is interesting because, yeah, there is a diversity, but they are not connected with each other. Yeah, and we also went into the metagenome of the lettuce to understand the relationship. And it is surprisingly, you found a lot of enterobacteria on the lettuce phylosphere. Normally, the people from the administration, for example, they only analyze pathogens on lettuce, but we could show that they belong to the natural community on the lettuce phylosphere, and that they are especially associated with the phylosphere and not with the root. Yeah, these are the functions. Yeah, you can do here everything and compare then the function, how it changed from soil to the phylosphere, and maybe it's nicer to go to this picture, because otherwise you need a lot of time to go into the detail of all these networks and on all these figures. But as I told you, we compared the soil microbiome, the rhizosphere microbiome, and the phylosphere microbiome. And in the first picture, you could see this rhizosphere colonization by bacteria. So, but we were not able to find any bacteria under the microscope for the phylosphere. But we saw the diversity in the amplicons and in the metagenome, but not by microscopy. And it took more than a year that we found the reason for that. The reason is that all the leaves are covered by a wax layer, and the wax layer has an antibacterial effect, so there's more or less no bacterium on this wax layer. In some cases we find single colonies, but maybe they were only from the air. So all the bacteria moved inside, and they live inside their endophytes of the lettuce leaves. And you can see here really a dense colonization below the wax layer, as well as here inside of the tissue, but you cannot see any outside. So that means we eat huge microbial diversity by lettuce, and first it was really a surprise that we eat so many enterobacteria with this lettuce. But after avoiding lettuce for 2 weeks, we start again eating lettuce, and then we developed the theory that it is a natural vaccination which we get by the lettuce eating, and maybe it's much more important for our health than to eat, because there are not so many minerals and so provided by lettuce. And we also published this as an opinion paper, and we will now go on to really show the relationship, because it's interesting. Our immune system reacts mainly with Enterobacteriaceae, so it develops by the contact of, especially of this group. I don't know why, but it seems to be important in the past. So, hope you will have a salad for lunch today, so that you will be healthy. I will conclude this, which is the biggest chapter of my talk here, because this is our main work, this basic work on the plant microbiome, and a small overview about the other things which we worked on, and which shows you this conclusion that diversity and networking helps to avoid pathogen outbreaks and is against pathogenicity. So we also worked with grapes, in vineyards, and we could show that the organically managed systems have a higher diversity than the conventional managed grape systems, which was a surprise for us. So, but it seems here that also a lot of natural biocontrol agents are enriched in these systems, and this was also in collaboration. We have Berg, and this is also not a university, but all the people working on the vineyards. Styria has 3,000 of these private vineyards, and they produce also very good wine, by the way. So it's not industrial produced, it's really private with their own. I think many Italians like this also to come to Styria and enjoy this. So it's really true that also inside of the grapes there's huge diversity, and a lot of our farmers, they produce a wine with indigenous microbes, which works well. And by the way, they are really important for the taste of the wine, because the microbes produce a lot of these specific compounds which is inside of the wine. So then we had also these studies about the wild type, and this is a similar study which I showed you for the lettuce. We have done with the sugar beet, and we found also that here breeding has an enormous impact on the microbiome, and it is really important for the breeders to integrate this in their strategies. And for a long time I have also a project with medicinal plants. So you know that the medicinal plants produce a lot of antibacterial, antifungal ingredients, and they have a very specific microbiome, and surprisingly, this was the highest diversity we ever found, associated here with chamomile and with Solano, what is Ringelblume? Calendula. And yeah, this is very important diversity, and there's also an interaction between the ingredients, between the bioactive ingredients for chamomile, for example, and the bacteria. And it could be that also one of the effects of the medicinal plants is their microbiome and not only the substances. And we had a collaboration with Cordoba University and with an institute, Agricultural Institute, about the vascular wild and olive trees, which is verticillium. You can see here the dry olives, and this is, yeah, this is in Spain. It is a really devastating, serious disease, not in Greek, because there the diversity is higher. So it comes from management practices. This was established there because it is correlation with the diversity of higher plants. And we also worked with bananas, with Uganda, and with this is our main crop in Styria, so the vine and the oilseed pumpkin, and also with strawberry. And so the conclusion is really that diversity associated with the plants and networking is very important for the health of the plant. And the other conclusion is that we have a specific microbiome. It depends on the plant species. For example, for the monocotyledonous plants, there's not so much diversity than for the dicotyledonous plants, and the specificity can be explained by the transfer via pollen or seeds, and by the secondary metabolites and the morphology of the roots. Yeah, these are several references about the specific plant-associated microbiomes. It started with cultivation, then with fingerprints. So this is a review of Connie and mine, because we had for a long time, 6 years together, project about this plant-specific effects. And now it was also in Nature for Arabidopsis, because you know that Arabidopsis is our most important plant. So then a short excursion to the intensive care unit. So I had a project with a clean room, a company which produced clean rooms and also those equipment for the intensive care unit. And they had so many problems with their sterility inside. And so first, I don't like the project, because I thought I'm working with plants and I will not go into this sterile environment. But now I'm really happy, because I learned a lot from these environments. And the most important thing what I learned was that the beneficials inside of this intensive care unit, they are coming from plants. So plants are a very important source. The phyllosphere of plants is a very important source for the beneficial bacteria inside. So, and we need these stable communities, as I told you, but we need it also indoor. And due all to this management and hygiene practices, it is more or less destroyed. And then you have only this network of the multi-resistant pathogens. And the only source for good bacteria is sometimes to open the window and let some in. And when we have done this study, it was also a problem, because we found all these plant-associated bacteria. And then they said, we cannot publish this because we are not allowed to open the window, but we are doing it because it's normal thinking to do that. And here, we have done it together with our people from the university hospital, and we found out that by their method, they only found 2.5% of this diversity. And they cannot assess all these bacteria which are indoor, but they are really important for our health. And what I found really interesting, because for a long time I worked with biocontrol and plants, the people are skeptical about this, and especially the farmers and the companies. And I thought if we publish this study about the hospitals, that they are so full of bacteria, so they will kill me. But it was completely the opposite. So this is here the response from the journals, and this is Kronenzeitung. So this is very low intelligent level, but they formulate the sentence: the fight of bacteria within hospitals. They were on the side of the bacteria, so this surprised me completely. Or the hospital need good bacteria, so it was so they see it much more than all the farmers which then we have worked. So, and we checked all the literature about indoor microbiomes and plant microbiomes, and we could really formulate our hypothesis that plant-associated bacteria are the source for beneficials. And now we also, here we also could show it for house plants, because all these brown dots, so these are bacteria coming from the plants, which are green, and landing on surfaces and in the air. So it is really a source for beneficials. And now we have this complete here, this interaction of the microbiomes, because now the people are focused on one microbiome, rice microbiome also. But everything is so well connected in our ecosystems. And here it was shown, for example, last year in Nature, that the people, the bacteria from the plant microbiome survive. They survive everything, the stomach and the gut, and they enhance our diversity. So therefore, if you eat the lettuce, it stimulates not only our immune system, it enhances also the gut diversity. So we have seen this link, and also for the human microbiome and the indoor microbiome, it was shown here in 2012. Per hour, we emit 10 to the 6 bacteria. So if we sit here together, we also exchange our microbiome, and at the end, you will not have my knowledge, so you will also have a little bit of the microbiome. So, and this was a link now from the microbiome to the plant microbiome, which I have shown you here. And by the way, there are a lot of links from the human microbiome to the plant microbiome. So first we thought that it is so unique. So, but now analyzing and understanding all these functions and ecological rules, we really see that there are a lot of links. And if you want to read something about the human microbiome, it is a really nice book about missing microbes. So about the human microbiome from Martin Blaser from New York City. So it's very interesting. Yeah, these are the similarities. So we have different microenvironments, so our skin, our mouth, our stomach, and our gut. Everything is connected. The plant, for the plant, we created these nice names: rhizosphere, phyllosphere, endosphere. So we have the early stage perturbations, which is well known for humans in the first 3 years. So then they get a lot of diseases, and also for the plants, it's well known in the first weeks. So there are a lot of dumping off, so-called dumping off diseases. And then we have a very stable microbiome over the whole active life. And then we have the senescent phase with a lot of enterics. So in both cases, these are the enterobacteria here. We have a host microbe co-evolution for everything. We have a vertical transmission of the microbiome for humans and for most of the plants, with exception of Arabidopsis. And so the human management practices cause a lot of diversity. This was very well shown in this Martin Blaser book, so that a lot of diseases which we now have, these chronic diseases, asthma and all these gut things, and so it really comes because we don't have these bacteria anymore. And together with his wife, he investigated the Indians in the Amazon and in Africa people, and they have much more diversity than we. So maybe in Italy it's still fine because you have good food, but the traditional Western diet, the American diet, is very, very, very bad. So, and yeah, the function is, there are a lot of similarities, and the composition is also very similar. And normally bacteria are the key players, but we should not forget the archaea, which are everywhere, also in plants. So we found a lot of archaea, by the way, in olive trees and in other plants. And also the fungi are there, and also the protists and so on. Diseases are often associated with a dysbiosis, and we don't know what was first, the dysbiosis or the pathogen. I think the imbalance. And we have this diversity pathogenicity relationship, which is well established, and the only importance for diversity, I think. And the microbiome interact a lot with the hormone system, also with our hormone system, and this should be the reason that a lot of young women cannot get pregnant because of these. They have an overproduction of hormones by their gut microbes. And we have also this host microbe rhythm relationship, which I like very much because it explains our problems with jet lag. And the jet lag is not the problem of our cells, it's a problem of the microbiome. And if you avoid to eat anything during the long flights, so then you will not have these problems with jet lag, so which is very nice. So at least I will give you some conclusions for biocontrol. So as I told you, for a long time I'm working in this field, and it completely changed also my mind about biocontrol, and I would now call it advanced biocontrol technologies or microbiome control, because it's not possible to use single organisms and don't take care. But sometimes it works. So here are our products. This is an endophyte, also fighting against D. dumella and bacterial pathogens on the oilseed pumpkin, which is an important disease, not so much the D. dumella, but it invades into the pumpkin, and then we have a lot of xanthomonas and erwinia, and they fermented the whole inner tissues, and then you cannot use them. We have here Salavida. This is by Sosson Pardena, so we developed it and then we give it to the company. And we have also the Rhizostar against verticillium wild and strawberry. But now we try also to use this knowledge from which we obtained from our basic research, and to transfer these ideas to new biocontrol approaches. And Christine has developed here such a new technique to catch from natural vegetation bacteria. So we isolate the whole microbial fluxion. For example, for mosses, it works so well. And then we treat the seeds and the rhizosphere and the phyllosphere, and then we look by these molecular techniques who is able to establish, and we were able to develop a very interesting cocktails of stress protecting agents. They come from the moss and ended up in sugar beet, and they are already in the commercial development, so it was very fast because it works so well. And this was due to the fact that we learned that the microbiome of sphagnum is so effective to protect the plants against abiotic stress, and it works easier than against biotic stress, because with the biotic stress you have a lot of more components, so you can use that. And you can also use the mining of metagenomic data set to find new substances and new enzymes, and to combine this knowledge with the isolation of microbes with all these functional approaches. This is what we are doing in the ASIP. And this is the last point. This is here one of the stress protection agent. This is Stenotrophomonas rhizophila. Here it is in salinated soil, and here it is treated with Stenotrophomonas. And I isolated this strain already during my PhD thesis, and for a long time I'm working with the strain. But in former times it was only basic research, because we were working for a long time about the differentiation of opportunistic pathogens, because they are close relatives to Stenotrophomonas rhizophila, which since the 80s they cause a lot of problems in hospitals. And after 15 years now it comes to another point, and because it was in a collection, in a culture collection with our Uzbekistan project, and there it works so well against these abiotic stress, because they have highly salinated soil. It is unbelievable that plants can grow inside of this soil, all these problems with the Aral Sea and Ammonium Surdaya system. So and now it was possible by transcript atomic analysis to detect the mode of action. In former times, I analyzed it for a long time by all biochemical and genomics and so on. And we found out, interestingly, that the reaction of root extracts, verticillium stress, and abiotic stress is the production of glycosylglycerol. We know that from our former studies, this is a very effective osmoprotectant, and it protects the roots against this salinity. And the other one is spermidine. So this was first a surprise, because I never heard that bacteria can produce it. But then we found that it is not only a very important substance protecting the human sperma against any stress, it's also produced by plants and by other organisms. And the Nature title was the novel anti-aging drug. So it is difficult to find a category, so it's in between a hormone and a protectant, osmoprotectant. Maybe anti-aging is good. So this is spermidine, and produce a lot of them. And also in these investigations, fortunately, we found that our biocontrol strain has a suicide vector working at 37 degrees, so that there is no risk. So I will end with this network from lettuce, because this was also the title of my talk, that this networking is very important for health and as well as for diseases, by the way. And analyzing all these networks, you can also find here, you can identify indicators of healthy plants, but you can also find indicators of diseased plants, and you can analyze the core microbiome. Here these are all our lettuce data, and this is a core microbiome for all lettuce plants, this for the phyllosphere, this for the rhizosphere. And we found out that pathogens as well as beneficials induce a microbiome shift on the plants. And I think this is very important for further analysis, to analyze these microbiome shifts, because maybe they are more important than other symptoms. So, and this is especially for Victoria. So normally we come from the ecosystem level and we analyze everything from the ecosystem, but sometimes we also go into detail, because I think it's very, very important. But this is not always easy. Here what we have done is we analyzed the interaction of Penibacillus and verticillium, and we found that they have a crosstalk via VOX. And if you put them on plates, and not together but with this hurdle, and then they started a talk over 14 days, and one produces antibiotic, the other responds then with this antibiotic, and it is so unbelievable. So if you have only two strains and you see how they interact, and then you have the whole ecosystem, only one microbiome, so only one rhizosphere, and you have 1,000 species, and then you can believe how they interact. Yeah, a lot of things to do. So this is my conclusion. But I really like also this detailed work from time to time to see that we capture only a small proportion. And yeah, I think this is my last word. Life did not take over the globe by combat, but by networking. This is the old woman which established the symbiosis, Lynn Margulis. And so this is our vision also a little bit, to combine our results for also not only for plant health, also for our health. And I hope that I could show you that everything is really connected, and that there are a lot of things which we have to do in the future.